Hey guys, sorry I'm a few seconds late. My camera decided it didn't want to be a camera today, to which I said, you need to be a camera. We have things to do because we have to talk about meiosis and we have to give a little introduction to heredity. So congrats, that's what we're going to do today. Thank you for giving up your Wednesday evening to talk about biology. I know lots of you have other things to do. Ooh, and I see we got some people. Natalie, Malin, Madison, and Shweta. Thank you for being here. My name is Jessica Nadsum, if you've never been here before. I do this a lot, and not just streaming. I am a 5th-year AP biology teacher. I am a certified AP reader, which means they have taken me into a room in a hotel and had me spend 8 hours a day grading AP tests. So I know what I'm looking for. I like to think I'm pretty good at it, and I hope I can share some of that information with y'all today. So, woohoo. But let's take a look at this thingamajig, this beautiful slide that I made you. I forgot to put my name on it. That was silly of me. As I said, my name is Jessica Nadsum. I have been doing this for a minute. And it's going to load. So just a heads up for you guys. We do have streams going on every week, Wednesdays, same time. So 8 p.m. Central. So if just go ahead and throw that on your calendar because you're going to want to know that. All right, let's move into the things you actually need to know. Do not forget, please, that we have a social media presence because we're millennials and we like social media just as much as you guys. So if at any point you're ever using your Fiveable materials, or if you have an idea for Fiveable, or if you just want to look at some of our content, feel free to check us out on Twitter, Instagram, YouTube, all those things. We have lots of stuff, and we're in the process of creating tons of more stuff. I personally am making lots of stuff. And so if you like the stuff I do on the internet when I'm talking to you guys, hopefully you'll like some of the other stuff that I'm making. So check us out. We're cool. We hope you think so too. What are we going to talk about today? So today, that is a lot of stuff going on that screen. I spent a long time on the internet finding all these GIFs, so I hope you're all grateful. Okay, so first things first, we've got to talk about meiosis. If you recall, last time we talked about the cell cycle. So now we're talking about meiosis, which is going to be for reproductive cells. You need to know how reproductive cells are made because then we're going to get into genetic diversity, which is important. And then we're going to get into heredity, which is also important. And I'm going to show you a bunch of photos from my family photo album because I am a genetic anomaly. And as a biology teacher, I find that a great way to make science relevant. So going back to this again, we're going to talk about meiosis. We're going to talk about genetic diversity as a result of meiosis because that's a question they love to ask on the AP Bio test. And we're going to talk about heredity. How do we change as we go through the generations? It's like, if you've ever played like the original Mario, like I said, I'm a millennial. I had the original Nintendo in the 90s, and Mario did not always look this well defined. He used to look like this. So how do we change? How do those traits get passed on, and how do they change over time? So without further ado, let's review real quick the cell cycle. We talked about this last week. I think it was last week. I don't know. There's a lot of Wednesdays. They all get blurred in my head. But you have a Nintendo 64. Okay, Dakota, I had one of those too. But there was one that was older than a Nintendo 64. Like these were the first cartridges that you would like have to blow on in order to get the cartridge to work. But very cool. I envy you. I don't have a Nintendo 64 anymore. So going back to the cell cycle before I got distracted by video games. Sorry, guys. I'm easily distracted. We talked about this last week, this cell cycle, and so it goes through this wheel of different phases. Can anyone remind me, what do we call G1, S, and G2 collectively? What do we call those phases? G1, G2, and S collectively. It's a long word. It's a fancy word. Think. Interphase. Very good. Okay, so all this stuff surrounded in orange is going to be interphase. And this is like the cell's main majority life cycle when it's just like chilling, doing cell stuff, growing, all that. And then we have the M, which is going to be for mitosis. And that's going to be when the cell divides off and makes new cells. You can see mitosis occurring here in this GIF on the left. And so it starts off one cell, and Then the chromosomes have to condense from the chromatin, and then it splits, and then you have new cells, and then they turn back into noodles called chromatin, and la la la. We've already discussed this, so I'm not going to spend a ton of time. But for anyone who wasn't here last week, I want to make sure we're all on the same page. So does anyone have any general questions about the cell cycle, mitosis? Three, two, one. All right, high five. Go team. Let's keep going. All right. So this is the part that most of my students struggle with just because, you know, oopsie, biology teachers are evil and biologists are evil. And we like to make big, long words that no one else understands. And when they're really important, we like to make another word that sounds exactly like that word to confuse people even more. That is a false story. I just made it up off the top of my head. But these words are easy to mix up. Mitosis and meiosis, they both start with M. They both have an osis. And they're both very critical to reproduction. There is a difference, though. So while both of them do segregate or separate chromosomes, and both of them do produce more cells, mitosis is going to be for creating 2 diploid cells. Meiosis is going to be for creating 4 haploid cells. So don't be like Clone Spider-Man. Like, don't get confused over these. It's all going to be good. In your toe, mitosis occurs. Madison, that is amazing. I need that in a banner in my classroom. I'm going to remember that then. But very good. Like I said, don't get confused like twin Spider-Mans about which these are. It's really simple, especially with the toe joke. But so mitosis, we're only going to make 2 cells, and those cells are completely identical to the original cell. So this is like, I'm trying to think of an example. So for example, let's say I get a paper cut. I hate paper cuts. Don't you guys hate paper cuts? So there's a little gap in my skin and something's got to heal that. I'm going to need mitosis to make new cells to cover that cut in my finger. That's what I use that for. On the other hand, meiosis, I'm only going to use very occasionally. In fact, women actually do most of their meiosis before they're born. Females are born with all the eggs they're ever going to have. They're not completely done, like they have to be cultivated, so to speak. But ladies, we have already done all the meiosis we are ever going to do, just so you know. You were born with all the eggs you've got. Whereas the males, on the other hand, do meiosis frequently because their genetic material is, to put it lightly, disposable. So meiosis, if you're a lady, you make an egg. That's an egg. If you're a dude, you make a sperm. The best way I can describe the egg and the sperm, think of the egg as like the sun. The sperm is like the size of the earth in relationship to the sun. Like it's tiny, it's disposable. Still can be done more frequently. Whereas females, these are massive cells. And there's not nearly as many of them made. And so they're a lot to gamble on. Plus, females will only release them every 28 to 32 days. Typically, there are obviously exceptions. Is that why women go through menopause? Yes. So Dakota, that's a good question. And there's lots of hypotheses. We kind of get into this with evolution, why they do, because most mammalian organisms, once they are no longer reproductively fit, their lifespan doesn't typically extend past that. Humans, for some reason, though, are very good at living for like 30 years past their reproductive years. That is females. Dudes can reproduce into their 90s. It may not be high quality, but they can. There's a hypothesis called the grandmother hypothesis, and it's basically saying that families and communities, because humans are community organisms, benefit from having a grandmother figure to assist with the children. And so women go through menopause because, and it's not like they run out of eggs. It's not like you have 500 in there and it's like, whoops, they're all gone. But eventually your body just stops producing the hormones that release those eggs. Does that make sense? Okay, very good. So when this process actually occurs, whether you're an embryo or a full-grown human male, you don't end up with something that is identical to the original. So like I said earlier, in mitosis, these are identical to the original cells, and they're supposed to be. If they're not, what does that lead to if they don't look the same as the original? If mitosis doesn't create the same thing as the original, what kind of disorder can you get? Cancer. Very good, Madison. So cancer is the result of unregulated cell growth. And that when that typically happens, it's like these cells are making copies so quickly that they'll miss things. So like the best way I can explain it is, if you've ever made copies on a machine before, you know, there will be like a high quality option, a low quality option. And the low quality option works a lot faster, but the pictures don't look as good. That's mitosis. You want the high quality answer every time. Meiosis, they don't look like the same as the other ones. You end up with 4 cells that have half the genetic material. Why? If I'm a product of a twin, does that mean I'm more likely to have twins? Dakota, that's a good question. It depends on a lot of factors. Like if your twin parent was fraternal or identical, and I think it skips a generation, but they also haven't really proven that. They're identical. I don't remember which one is genetically linked. I would have to Google it. I can do that if we have time at the end, though, so remind me of that. But so kind of going back to this meiosis, you end up with only half of the genetic material as what you originally started with. Why is meiosis trying to produce cells that have half the genetic material? We just talked about cancer and how we want to have all of the genetic material. So why does meiosis make cells with half the genetic material? To reproduce? Elaborate on that, Madison. So like, why can't they reproduce with all their DNA? Like, why can't I give all of my DNA to my offspring and my husband give all of his DNA to our offspring? Why am I only allowed to give half? 2 halves make a whole. Very, very good. So when you've got, get my markers here, because both parents donate 1 gamete. Very good, Natalie. So this is not drawn to scale, but when you got an egg and you got a sperm, they each have half. That comes together plus, I know this is backwards, equals a whole. Ta-da! Sorry, y'all can't see that. I've got a ring light, which is really helpful because the lighting in this room isn't good, but it makes it really hard to use that whiteboard. But yes, 2 halves make a whole. Each parent gets 23 chromosomes. Very good. Both parents donate 1 gamete. Exactly. So like, if I were to have children with my husband, I could give half of my genetic material. He could give half of his. I may be like, but I really like my genes and I just want a mini me, but can't do that. And that's probably a good thing. So let's take a look at this next slide real quick. So got a lot going on here. Meiosis is all about reproducing. And when I think about that, I like to think about 101 Dalmatians or like piles of baby ducks because they're so cute. And baby animals. Who doesn't want baby animals? So meiosis is going to go through this exact same process as mitosis, but how is it different from mitosis? How is the process of meiosis different than mitosis? There are 2 phases. Very good, Madison. So in meiosis, I've got this meiosis 1. And meiosis 1 looks an awful lot like mitosis. And then I'm going to have a meiosis 2 phase, and that's also going to look a lot like mitosis. The only difference here is how those chromosomes are going to line up, because they don't actually line up in any way, shape, or order. So like if you've ever been in class and they tell you, everyone line up by alphabetical order, and then you all shuffle around like, okay, well, my name is A. Well, yeah, your name is A too, but mine is like A, you know what I mean? So those chromosomes don't do that. They don't line up like, okay, I'm taller, I'll go here, you go there. They don't do that. They go where they want. Very good, Natalie. It's called independent assortment. They just line up wherever there's room. And as a result, that's going to create some variation. So like, if you look at all the Dalmatian puppies, for example, the Dalmatian puppies are not identical. They all have the same basic features. They're white, they got black spots, and they're so cute. But their spots aren't all in the same place. And if you watch the movies, the personalities are not all the same. You got like the hungry one and the cold one And there's a bossy one, and I think there's 98 others, but I don't remember them all. So one other key thing I need you to know about meiosis, because this is something they love to bring up on the AP Bio test, is the homologous chromosomes. So this vocab, I will be perfectly honest with you, this is another example of where biologists made a bunch of words sound alike just to confuse people. And it drives me nuts because I don't think I fully got it until college. So let's take a look at this here. So in meiosis, you've got homologous chromosomes. Okay, they're there. They're doing what they're supposed to. But they replicate. And when they replicate, you have these sister chromatids. So sister chromatids is like here when they're crossed over. But in meiosis, they do something cool. They will actually sometimes cross over with the chromosomes next to them that are in that same homologous chromosome. And so you can see here like how we've got blue and red that have swapped. And that'll create more genetic variation too. So you may only pass on, say, mom's chromosome, but because of crossover, you now have a little bit of dad's gene, even if we didn't send on dad's chromosome. So genetic variation. We like genetic variation because if everyone looked the same, it'd be really annoying. And then we're going to go on, separate twice, and at the end we have these gametes. They each have only one little chromosome, and they're going to combine with another gamete that's going to have more chromosomes. Fun fact, though, when human females do this, do we have 4 viable gametes? No. Very good, Madison. So what happens here? So like in dudes, males, very good, Natalie. We have 3. Madison, so not 3. You got it a little bit backwards there. We actually have 1. So what happens, we have 1. Yes, Natalie. So what happens is, like guys, like I said, in males, any male mammal, they have much more frequent reproduction through meiosis, basically disposable gametes. And so they will make 4 at a time for each process, and that's fine because these are small and they're not meant to live very long. I think sperm can live at maximum, if in optimal conditions, like 8 days. Whereas female eggs have to survive in an organ called an ovary for up to 40 years. They have a long time to go. And so what happens is in females, when you're in utero, when you're a little embryo, you go through mitosis. And yes, you make 4 gametes. But they basically play a big game of survivor and realize none of us have enough fat and materials to I'm going to make this bigger so y'all can see, sorry. But basically, when they're going through meiosis and they've got these 4 gametes in the female system, they basically realize not all 4 of us are going to make it 40 years with the amount of materials that we have. But if they all give all of their materials to one other, so one of them gets massive and can survive, and the others are just little dots with nothing left, and they all will die. But evolutionarily, that big one is much more likely to survive for 40 years as opposed to these 3 that wouldn't survive, those 4 total that wouldn't survive because they wouldn't have enough materials. They wouldn't have enough fats, proteins, things like that. Does that make sense? Okay. Cool. I'm trying to think. Does anyone else have any questions about meiosis? The AP Bio guide says you do not need to memorize the specific steps. Please don't memorize the specific steps. Don't freak out about that. The main thing they want you to know is the mechanisms for increasing diversity, which we're about to get into. So any questions? All right, I'm gonna hop on along then. Okay, so now we have our much more in detail chart of meiosis. I'm gonna go pretty quickly through this because like I just said, don't memorize all of this. 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 It's great if you can, like, woohoo you, I bet you can memorize like 50 digits of pi as well. That's great. But in practice, you won't need it, at least not right now. So, but the points I want to emphasize here are crossing over and independent assortment. So whenever these things go out of interphase and go into the first part of meiosis, that crossing over happens here in prophase, and that's where the chromosomes are still in their sister chromatids. They're crossed over. And that's when they will actually start. You can kind of see it here, how it'll have 3 ends blue, 1 end pink, or 3 ends pink and 1 end blue. And that's when they've exchanged their genetic material. So although this is primarily, since it's blue, I'm going to be stereotypical and say it's male, but that blue doesn't mean male. You know what I mean? But since this one's blue, I'm going to say it was the male gamete, or it's the gamete that was inherited from the male parent. But it crossed over, and now it has a little bit of the gamete that came from the female parent, the pink. And so then those things separate. And when they separate, like I said, they are strong, independent chromosomes. They're not just going to line up like, okay, you're tall, you're short, you're tall, you're short, boy, girl, boy. They don't do that. They're chaotic, and they go where they want. And so as a result, when they're separated here, it's not like we're getting all of them on one side. So then we go through this again, but this time all we have to split up is the sister chromatids. All that's splitting in meiosis 1 is the homologous chromosomes. That's the same thing as what happens in mitosis. Meiosis 2 is when we start having those sister chromatids get split apart. And that's when you end up with these things, teeny tiny little DNA, and then you get those 4 cells. Whoop, whoop. Okay, so let's harp on that genetic diversity. For the record, to be fair and equal, I tried to find another GIF of this, but Bernie just says it best here. Our diversity is one of our greatest strengths. I tell my students this constantly, and Bernie's kind of freaking me out, so I'm going to make him smaller for a second. It is imperative to the survival of all living species that you have diversity. Has anyone here heard of a thing called inbreeding? That's rhetorical. You don't have to answer. Most of us know what that is. Inbreeding is the result of a lack of genetic diversity. It's when siblings marry siblings or cousins or whatever. Empires are really good at it. So like the European empires, the Egyptian empires, President Roosevelt does. So inbreeding, most of those dynasties are either weakened or no longer existent. For example, the Egyptians, because they kept marrying their cousins and siblings. That was the norm, and it was meant to keep the bloodlines pure, which in theory, if you don't know what DNA is, like, whatever. But after a while, kids start being born, and they have serious problems, whether it's deafness or blindness or developmental delays. That's a result of a lack of genetic diversity. So, and then you can look at this also in purebred dogs. So I personally have a mutt. I love my mutt. She's so cute. But one of my favorite things about this my mutt is she's genetically diverse. So I don't know if y'all can see her. There she is. That's Ellie. Isn't she cute? She's a lab mix. We think like pit bull or something. We don't really know. But Ellie is genetically diverse, which I think is good because a lot of purebred labs. Has anyone here ever had a Labrador Retriever before? What's a common problem in labs and big dogs in general, especially when they're purebred? Epilepsy? Is it a purebred dog? Yeah, that can happen. What's another common issue with big dogs? Typically occurs back here in their hips and their legs. I just have stuffed animals all around my house. Arthritis is common. Another really common one is called hip dysplasia. So hip dysplasia is the hips aren't lining up right. I don't know the exact mechanics of it, but it can be very painful, and it can happen really early for dogs, which can lead to a lot of expensive problems. And it's really common in purebred dogs because they have, yours is deaf. Purebred dogs have a lot of issues compared to mutts because they have low genetic diversity. If you're getting a purebred Chinese crested hairless dog, that dog can only breed with other Chinese purebred crested dogs, hairless crested dogs. And as a result, there's not a lot of genetic diversity. And when there's less genetic diversity, if there's a problem, there's not extra genes for them to rely on. Dakota, yours is deaf, but a mutt, hey, mutts, the mixed breeds, they have issues as well. Everybody does, don't we? They just happen at a much lower rate as compared to purebred dogs. So I'm sorry to hear that, though. Poor thing. Is she all white? Is she like an all white dog? No? Okay, I don't know what it is then. Deafness is really common in dogs that are all white. There's a linked gene there. It's basically called a double merle or something like that, I think. We don't have time for that. We got to go. She's a black lab. Yeah, I have no idea. Not a clue. I thought you said she was a mutt, though. Now you're saying she's a black lab? I'm so confused. Oh, well, it's okay. But so like I was saying, as Bernie was preaching, our diversity is one of our, that's what the shelter said. I bet she's a black lab mix. Okay. Anyway, diversity is one of our greatest strengths. Evolutionarily, we need it. There's a reason we don't still look and act like cavemen, because we've adapted to be better. But meiosis promotes that genetic diversity through the act of crossing over. So you can see here the homologous chromosomes. They want to dance. And then they switch. And then they cross over. And so we get that genetic diversity because all 4 of these lines are going to get split into separate cells by the end. And then we have our, and so you can see here, there are 4 genetic arrangements. Here we have all the blue ones. Here we have all the red ones. Here we have blue and red. Here we have red and blue. So there's 4 different possible mix-ups in that image alone of how meiosis can go. And that's how you get all the different chances of having a certain kid. That's what Punnett squares are. Because Punnett squares, it's 4 boxes. Figuring out what is the chance of my kid having this. Got it? Cool. Let's go. Dun, 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 dun. All right. This is my favorite part of the lesson. So I teach biology. I have for 5 years. Every year I show these pictures. I will say I got married last year. So these are all my wedding pictures. So please put up with it. But I paid a lot of money for them. So I must show them off to the world. Plus, this is real. So these are my wedding pictures. This is me. Hi, me. Oh, it was such a good day. It was really cold that day, though. But so I want you to look at these pictures. The top one and the bottom ones. Which one do you think I'm standing with my brothers? I have 2 brothers. Which one of those do you think I'm standing with my brothers? Oh, hey, thank you. There was a lot of hair and makeup that day. It's not how I look every day, as you can see. Which ones do you guys think are my brothers? Which ones look like my brothers? Bottom picks. Come on, more y'all chime in. This is which one you think. There's no wrong answer. This is a poll. Dakota? Madison, don't overthink it. It's okay. This must be a trick question. So first, okay. Wait, what was the question? Okay, I was asking Dakota, which of these 2 pictures, the top or the bottom, which one do you think is the one where I'm standing with my brothers? Top. Why do you? Okay, I'll go with it. So I show this to my kids every single year. The second, you look the most like the bottom. Thank you, Madison. Okay, good clarification. So I show this to my kids every year because I think it's hilarious. Because when we're getting into heredity, it's all about family trees. How are you related to people? And I want to make clear, as I was talking about genetic diversity, it doesn't matter what you look like. That's not necessarily your family. For the record, I'm not adopted. Like these are my blood brothers. Like I was present when they happened. So this top one, these 2 boys that look nothing like me, those are my genetic brothers. I am a genetic anomaly. You're right. I shouldn't have given that away. These are my brothers on top. The 2 boys in the darker suits. I'm the older sister. This guy here on the left in the black tie, that is actually my husband. And this is my brother-in-law. This is my family. This picture's like 5 or 6 years old. And for some reason, I didn't know how to draw in eyebrows yet. So please just ignore that. But I show this every year to be like, oh, hey, thanks, Natalie. I phenotypically, physical features, at first glance, I don't look a ton like my family. And people see it and they're like, how? I don't get it. What is going on? I know. I'm like the mailman's child. Thanks, Dakota. I appreciate that. I've never heard that joke before. No, my mom would take me to the grocery when I was a baby, and people would assume she was the babysitter. Like, I don't look like my mom. My brothers, as they've gotten older, you can tell we have a few of the same facial features, but like just from looking straight at us, we don't look a lot alike. Now, that being said, Dakota, you said I look like the mailman's child. So this is a picture of me and my father when we were both like 20. So my dad is this one. This one's me when we're the same age. So unless my dad is the mailman, I'm not adopted. We're good. But how does this happen? Like, because we've all learned about dominance and recessiveness in genes, right? So like you have dominant genes, those are typically going to be phenotypically the darker genes in terms of more melanin in the hair, the skin color, the eyes, things like that. And my dad even has dark hair. He does have blue eyes though, and I have blue eyes as well. And my mom is half Hispanic, and those genes went to my brothers. How did that not get to me? That's a Southern saying when someone really looks nothing like the rest of their family. Oh, I know what it is, Dakota. I just think it's funny because I look nothing like the rest of my family. But this is a picture of my dad and I when we're both about 20. And we look exactly the same in that photo. It's kind of scary. But so how does this happen? How does heredity work in this way that you look so much like your family? Or maybe so little. Like, for example, do you guys feel like you look mostly like your parents, or do you look maybe like a blend of your parents? What do you guys notice in your own family photos? I know, it's true. You look like your brother, Madison. You look like your mother. I'm sorry, I can't read. I still have my contacts in and they're blurring. Okay. So sometimes we look a lot like our family members and sometimes we don't. And I mean, there's other genes, obviously the mental ones. Like everyone tells me I act like my mother. So when we're establishing these family trees, it's really important when we talk about heredity to understand we're not just looking at those physical traits. And even if we are, they aren't going to be the same because we have genetic diversity. You have different things, and that's good. People say I look more like my father. Mm-hmm. Does anyone else, though, like they tell you, oh, you look just like your great Aunt Linda or something, and you're just like, I don't know who that is. What I will tell you, if you want to know who you look like, you need to find pictures of them when they're the same age as you. It's really hard to tell when there's like a 20-something year age difference, but if you get a picture of you two when you're the same age, it's a lot easier to tell. Free tip. So let's keep talking about this heredity now that I'm done sharing all these details about my family and putting my family portraits and wedding photos all over the internet. So going into this about genetics, so the parents have 2 chromosomes, but they only donate 1, the gametes. Those gametes then can either become eggs or sperm, depending on if it's the male or the female, and they can then be fertilized to create an embryo. And that embryo may grow into a fetus, which may turn into an infant, which may turn into adults. So, woohoo. So in this intro to genetics, I really want to bring up a couple different terms. Again, dominant and recessive. When we're first talking about genetics, those terms are going to be very important. At what stage is the gender determined? So that's a really good question. So I'm trying to think of the best way to do this. So are you guys aware of sex chromosomes? No, I'm just going to tell you about them for those who don't know. So you have 23 pairs of chromosomes, 23 from mom, 23 from dad. Very good, Dakota. And so, but your 23rd pair, you have either XX or XY. These are backwards, I know. You can have XX. I'm going to make this bigger. The rest are autosomes. Very good, Dakota. So you can be XX or you can be XY. If you're XY, you're a dude. If you're XX, you're a female. You're a lady. So you get those chromosomes when your sperm that creates you is fertilized. So I'm going to ask you, Shretha, at what stage is gender determined? When is the gender determined or created, basically? Anyone can answer. Is this something that happens like the day before the kid is born, the day the kid is born? Is this something that occurs like the moment fertilization happens? What do you guys think? During pregnancy. It would be before fertilization. Exactly. So it's the moment that egg and sperm come together. Because the egg from the woman, no matter what, very good, fertilization, the egg from the woman is going to give an X. Is it gender or sex? Okay. So I will tell you in biology, we just say sex. Gender is not a biological concept. It's not anatomical. Sex, that's XX or XY. Yeah, very good. So females can only give an X. Like, I'm a lady, I can only give an X. That doesn't mean I'm only going to have daughters, though, because my husband can give an X or a Y. Remember, you give half your genetic material, so he can give an X or he can give a Y. If he gives an X, I'll have a girl. If he gives a Y, I'll have a boy. So the moment that fertilization happens, those two gametes come together, whatever they are, that's when it determines this is a boy, this is a girl. Now, that being said, like I know we have ultrasounds and all that today. You can't tell from an ultrasound what the sex is until it's like 2 or 3 months in, because that's when people usually go in for that ultrasound. And that's because around 2 to 3 months, about 10 to 12 weeks. So boys, I hope I don't blow your mind here. For the first like 10 to 12 weeks, everyone anatomically is female. That's why we have some similar anatomy, even though boys don't necessarily use it. But we're all anatomically female for the first 10 to 12 weeks. But around week 10 or week 12, somewhere in there, this Y chromosome, if you're a boy, turns on and says it's boy time. And it activates testosterone and those other things that produce the male anatomy. That's when you begin to exhibit the male sex characteristics. But the fact that you're going to be a male is determined like at conception. Very good question, though. Thank you for asking that. Okay. And so once this is all passed on, you have all these different options. Let's talk a little bit about Punnett squares and this guy Mendel. You get. Oh, when does gender start showing? Okay, gender starts showing at like week 10 or 12. Or sex starts showing at week 10 or 12. Exactly. Sorry, I got backwards. But yes, gender starts phenotypically being displayed around week 10 or 12. Yeah, it's okay. Hey, guys, I mix up those words all the time. I am not an OBGYN. I am not a doctor. So I don't have the habit of saying those terms as often. We only have this unit once a year in my class. Okay, so any questions more about all of that? The basic intro to heredity and even about male-female sex and how that is determined. Okay, I'm going to go ahead and go on. Got a little bit more here. Okay, so let's talk a little bit about this guy. His name's Gregor Mendel. If you've had biology before, you've probably been told he was a monk in, I believe it was Austria. And in addition to being a monk, he was also the gardener and the cook. And he liked math. He lived in a monastery or an abbey, so he didn't have, he had a decent amount of free time, it sounds like, because he was just walking through his garden and was like, those pea plants look different from those pea plants. And these pea plants look different from these pea plants. But they can only be this or that. Long story short, from there, he figured out which traits were dominant, which traits were recessive. And he developed these Punnett squares that would calculate what are the chances of a certain offspring occurring. And so you have these 4 boxes. Each of those boxes represents a possible combination of gametes. So that's why you have 4 boxes. When you see those dihybrid crosses, that's when you'll have the massive Punnett squares with 16 boxes. That's because you have 2 traits. And there's 16 combinations of those particular chromosomes. Okay. Again, going back to this, I'm going to make this a little bigger for you guys right now. So in this case, which one do you think is dominant? Which phenotype is dominant? Purple. Very good. Big B, big B. Okay. So, and to be clear, phenotype is how it physically looks. Genotype is going to be those letters, the big B, the little b, the two little b's. And so we're going to use those two words very often in biology. They're going to ask you a lot about genotype, which is the genetic code, that big B, little b. What is it called when you have two alleles that are the same? That is, they're both capital or they're both lowercase. What do we call that? Homozygous. Okay, very good. And they can be homozygous dominant if it's like both big B, or exactly homozygous recessive if they're both little b. What do we call it when they have two different alleles? So a big one and a little one. Heterozygous. Very good. Okay. It's not going to be recessive. Recessive is only going to occur when we have two little b's. Heterozygous is when we have one big B and one little b. So how many, for example, how many heterozygous organisms can occur through this Punnett square? 2. Very good. 2 out of 4, 50%. So there's a 50% chance that the offspring will be heterozygous. What is the chance that the offspring will be homozygous recessive? 25%. 1 out of 4. Very good, guys. Great job. Ooh, y'all are so smart. Okay. Oop. Why are you freezing? Oh, that's why you're freezing. Okay. That is my basic intro to heredity. I have several minutes to answer your questions. So, what questions do you have? Ask me things. I know someone had one earlier. My class is actually, yeah. So my class is actually in unit 6 right now. We did ecology first and then did all of them. So we're in central dogma right now. So you're in natural selection then. So this is, Dakota, you've done this before. So that's good. What kind of FRQs could be asked about heredity? So when I developed the test for this for my kids, we're still in photosynthesis. That's okay. So you go different orders and different speeds and things like that. You're in 6. Oh, Dakota. Okay, we're in central dogma 2. Okay, Natalie, I'm focusing on your question. I apologize. What kind of FRQs could be asked about heredity? The heredity FRQs, so I'm trying to remember when I made this test for my students, because when they took the test for heredity, it was also their semester exam. So I had some other stuff mixed in there. So actually, if y'all will be real patient, I'll just get on my Google and check real quick, because I've got my old test. I'm not showing you my test, to be clear, but I need a refresher on what the topics were that I did in there. So I will check that for y'all real quick. Chi-squared, I want to say I did ask a chi-squared. Punnett square problems are highly likely, but they're not just going to ask you, what is the chance of the flower being white? They're going to ask you, what is the chance of the flower being white and the flower producing another white flower? So it's very technical. It's very long, and at least in the one long question you will have over it. And additionally, they also talk a lot about the cell cycle and cell cycle in terms of meiosis. With meiosis, like I said earlier, you don't want to know the parts about the actual steps of meiosis. What you want to know is the specific mechanisms behind how crossing over and the law of segregation work. That's going to be the things they ask you about. They're going to be like, what is the impact of crossing over these genes? And you're going to be like, it is this, because I studied. Okay, so I'm pulling up my old test now. Give me just a sec. Again, I'm not showing you guys this, but that's a really good question. I'm trying to remember what I put on there. So when I did heredity, the FRQs that I gave, that my students loved so much, and I have the world's slowest internet. today. Okay. Come back. Okay, so the first one I did, it was about genes, and this one also talked about, so this is a little more advanced. We didn't cover this in the introduction to heredity, but keep in mind, this is not just going to be Mendelian genetics. It's not just going to be dominant, recessive. You also get into that non-Mendelian genetics, which some of you may have had already, where you're going to have the codominance and the complete dominance. You're also going to be looking at phenotypic plasticity, which is when organisms change their reproduction based on their environment. So for example, if temperatures rise, all of the alligator babies will be female or something like that. So I would understand those and then apply those to Punnett squares. So it'll be like, an alligator has babies and it's 2 degrees hotter than it normally is. What are all the babies going to be? Well, those babies produce normal babies, and it's a lot of that. So you need to really understand how the environment affects phenotype and phenotype, how to apply that to Punnett squares. It is going to ask about fitness because all of biology is about fitness. So how does something be fitter or more likely to survive and reproduce based on the way it is reproducing? Let me look here at this next one because I have 3. Oh, one that I had was comparing mitosis and meiosis. So again, you didn't have to have the stuff memorized. But what I will say from that, you need to be able to compare and contrast them. You don't want to like list out prophase, metaphase, all this, but be able to tell the difference and what's the same about them. And then the last one I did, we did chi-squared at the same time as heredity. Your teacher may do it differently. Oh my goodness, Madison, good luck. But yeah, chi-squared was something we did when we did heredity, and they may ask you to do a chi-squared problem about a Punnett square, which basically means you are having to look at a Punnett square and say, is that Punnett square likely to occur that way? So, panic. It's really not that bad. I love chi-squared though. I think I teach it later this month. And then there's not really a ton on mutations here at this point. Like everyone always thinks, oh, mutations in heredity. We touch on those. We touch on chromosomal mutations, abnormalities. And those are the result of meiosis not working properly. But I wouldn't know like all the stuff about mutations yet, because that's more in the central dogma unit when we talk about the DNA and how it doesn't replicate properly, which leads to a mutation. Very good question, though. But that's all I can tell you. Okay. Ask me more questions. You got me for like 10 more minutes, so I am your walking, talking dictionary of biology. Someone had a question earlier and I asked them to hold it till the end, but I don't remember what that question was. Oh, the identical twin one. Hang on. Are twins genetic? So this is something you guys can do. It's called Google. But what I will say about Google is not everything you read on the internet is true. So make sure you're looking at like a relevant source. Okay, so it says here, having identical twins is not genetic. So sorry, dude. Just because your parents are twin doesn't mean you're going to have a twin, which you may think is a good thing, may think is a bad thing. I don't know. Okay. What other questions do you guys have? With something like Huntington's disease, we should justify the idea that dominant traits aren't common because dominants just mean that one allele is expressed over the other. Is there something else? So Huntington's disease isn't common because people with Huntington's disease, this one's tricky. It is a dominant thing. So it should be common because if you think about it, typically dominant traits are expressed more than recessive ones. So like my family, they have all the dominant traits. So technically it's more likely for the dominant ones to be expressed, and Huntington's is a dominant disorder. The reason Huntington's isn't very common is because the likelihood of surviving and reproducing with Huntington's is not necessarily very high. That being said, the disease does occur later in life, unfortunately, often after they've had children, so they don't know if the children have it or not. But usually once they start tracking it from the family, there will be some genetic counseling to prevent that reproduction if it could be harmful to the child. Does that answer your question, Natalie? I'm sorry, that was really long-winded. Yes, awesome. Okay, more questions. Ask me things. It is 7:51 my time. Happy to hang out for a minute. Five, 4, 3, 2, 1. All right, y'all. If y'all don't have any more questions, then I'm gonna get ready to go to bed because I wake up really early. So I hope this was really helpful for you. I see I've still got like 5 of y'all left here. If at any point you have any other questions, I've told y'all before, like you can find me on Twitter and the Fiveable teacher page, creator page, whatever we call it now. Feel free to reach out. Happy to help. Thank you for coming, guys. I hope y'all have a great rest of your week, and I'll see y'all not next week, but the week after. See you soon. Bye-bye.